This third session, the invited speaker is Dr. Abhay Phulke. He is working as a senior scientist at National Institute of Oceanography, which is the regional center of Mumbai. He is very young, very energetic, and very enthusiastic scientist, son of our Vidarbha region only. Earlier, he had served in Niri also for a few years, and today he has decided to deliver his presentation on plastic pollution in the marine environment, implications, solution, and the biomitigation strategies. Now, I would like to request Mr. Joel Kerketa.
Yes, sir.
Please introduce continue his brief introduction.
Okay, sir. Am I audible now?
Yeah, you are audible.
Okay, sir. Good afternoon, all of you. Most welcome in the third session. I am Joel Kirketta. Thank you, Dr. Mesran, sir, for giving me opportunity to introduce a senior scientist, a dynamic personality, Dr. A. Phulke, sir. He has completed his MSc and PhD in microbiology in integrated to CO2 and published more than 30 papers in international and national journals. He received many awards for his scientific contribution by government and private body, and also nominated for a prestigious CSIR Young Scientist Award in 2015. I think now, without wasting any time, let's listen to the thoughts of a person rich in experience about the plastic pollution and solution. Once again, we welcome him. Over to you, Mesram, sir.
Dr. Abhay, now you can start your presentation.
Okay. Is my slide visible?
Yeah.
Good afternoon, everyone, and thank you for your kind introduction, Dr. Joel, sir. Well, as you are aware about the environment, and the ocean is the major part of the environment, so-called the marine environment. So I'll be presenting today on the topic plastic pollution in the marine environment implication solution and the biomitigation strategies. Just last month, the United Nations has given the theme for the World Environment Day that beat the plastic solution to the pollution, plastic pollution. So one of the vice chancellor from here in Navi Mumbai, Chhatrapati Shivaji Maharaj University, they have invited me for the talk there also on this plastic pollution because on the marine aspect, our organization is working and taking very seriously is considering the government new policies like blue economy and others. So before I proceed with the actual topic, I would like to introduce about my the organization that what we are working on when it is established. Our institute is the National Institute of Oceanography with the mission to continuously improve our understanding of the sea around us and to translate this knowledge to benefit all, which is founded in 1st Jan in 1966. We have a headquarter at Donapola, Panji, Goa. You can see here the headquarter building, and we have a 3 regional center. One of the regional center where I am working is in Mumbai. You can see the down here, and the other one is in the east coast, that is Vizag, Vishakhapatnam, and the third one is in the Cochin, Kerala. So working in the ocean, we should have the research vessels also, the research ships. So we have 2 India's biggest research ship, which is well equipped with all the instrumentations and the different laboratories, microbiology, chemistry, physical oceanography laboratory, geophysical oceanography laboratory to just address the process related to the climate change and the other things. So here the first one is a research vessel RV Sindhu Sankalpa, and the second is the RV Sindhu Sadhana. The endurance day is also given that it can be in the ocean for the 30 days RU Sindhu Sankalp, and the Sindhu Sadhana for more than the 45 days. But recently, we have conducted for the 90 days cruise, and the 1 day expenditure of this cruise is 25 lakhs, considering all equipment, crew members, and the other requirement for the oceanographic study. And it is well equipped with the CTD system also, conductivity, temperature, and depth, which is very robotically go to the ocean. It can collect the different level of sampling or different level of water samples, and the bottom up to the 8 to 10,000 meter. That means 8 to 10 kilometer depth sediment it can collect. So it has the winch and the grabs like that to collect it. So it is a very sophisticated and advanced research vessels that our institute purchased. And these are the captain with me who run these ships, the executive captain and the captains, just to, wherever we want to collect the water sample, sediment sample, or wish to see is there any oxygen minimum zone where the oxygen is less. That's why the fish mortality will be more. So this process, biogeochemical process, we is to study using these research vessels. And before starting the once we on board to the ships, so we have different tips like 10 to 20 scientists and the students. So is to give the safety drill to the all students and the researcher on board, because in night walking around the ship will be dangerous. We always say that walk with the two people, and the other precautions also require if the ships get the fire, then what are the precautions we have to take. This all safety drills first before the sampling is given to the all the research scientists.
Just a minute,
And
Doctor
this is
Abhay,
yes, sir.
I'm sorry to interrupt you, but slides are not moving.
Oh,
Slides are not
is it
moving. Now it
now?
is
Now,
moving. Now it is moving.
okay, okay. So,
Please show
can you
some
see the seat?
earlier slides also.
Okay,
Please
okay.
show the earlier slides also. Yeah.
So these are shall I repeat? This is the three regional center our institutes purchase. One is in the Cochin, second is Vizag, and the third one is the Mumbai, where I am located. I am heading here microbiology and biotechnology division. And the next one is these are the two research ships we have, Sindhu Sankalp and the Sindhu Sadhana, which is well equipped with all the instrumentations and everything. The 1 day expenditure for this ship is more than 25 lakhs, as the sample is very precious. We need to have the permissions from the all government bodies like custom, marine police, port officer, and everything, as we used to go into the international waters near Mauritius, sometimes in the Somalia types. So Somalia sampling is always dangerous where there are pirates and all that. So we used to take the lots of precaution while sampling also. And this next slide, these are our ships captains who are taking care and running the ships. And the next one, very next one is this is the group, the different, the one who used to be the leader. So I led one of the crews. This is the photo represent the safety drill of all the research scientists before starting the sampling on board on the ships. And this is the CTD laboratory present in the ship only, where robotically we can collect by just watching on the monitor and giving the command on board in the ship. We can collect the sample of depth more than 8,000 to 10,000 meter. That means 8 kilometer to 10 kilometer down the depth of the sea. The water sample and the sediment we can just select, and there is one monitor also. We can see whether the winch and the rosette sampler is coming up at what level and when we has to go and collect the sample. Everything is automatically, and we had to just run the command and see. And while just commanding or giving the instruction to the computer or the software, we can see the different changes in the dissolved oxygen, conductivity, temperature, that graphs and everything will get on the monitor based on the depth of the water. So what does the marine environment actually includes? So here we can say आप लोग में से भी बहुत जब मुंबई या कहीं कोस्टल में गए होंगे तो बीचेस देखे होंगे, जो रिक्रिएशनल बीचेस होते हैं, और वो तो जो हाई टाइड लाइन होती है और लो टाइड लाइन होती है, here we can see the high tide line and the low tide line. इसके बीच के एरिया को इंटरटाइडल जोन बोलते हैं, जहाँ पे बीच होता है, रिक्रिएशनल एक्टिविटीज होती है. बाद में कंटिनेंटल शेल्फ होता है, और बाद में यहाँ पे आप डेप्थ देख सकते हैं. और उसके हिसाब से सबसे स्मॉलेस्ट जो फाइटोप्लैंक्टन्स होते हैं, फिर फाइटोप्लैंक्टन को खाने वाले जू प्लैंक्टन्स होते हैं, देर आर स्मॉल फिशेस, बिग फिशेस, sperm whale, octopus, and lots of, more than, as you know, that there are report more than 95% of the earth diversity is in the ocean, and only 5% of the biodiversity, marine biodiversity, is so far explored, and 95 is 8 too. So there is a huge scope as per the biodiversity management, biodiversity concerning with the aquatic and the marine life, and bioprospection from the marine drugs concern. So there is a large scope in the marine environment that we can explore. So these are some fishes, and we can see here the oyster having the polychaete, a gastropoda, and small fishes that we have collected for pollution studies in the ocean. And we are just looking for the heavy metal, pH, and we can say that even the microbial pollution in terms of fecal coliforms and the total coliforms, and some of the coliforms like bacteria like E. coli, Klebsiella, and the Streptomyces faecalis, they contain different types of toxin genes, which is harmful to the environment, aquatic environment, not only aquatic, to the human being also. It's consumed by the fish, and fish obviously consumed by the human being. So this is the general idea about the marine environment. Now let's move to our topic, that plastic, everybody knows it's creating the havoc everywhere, and the global commercial production of plastic was 2 million in 1950, and in the 2019 it went to 368 million metric ton, which is a very huge. So it is projected after a couple of years it will be 445.25 million metric. So how to deal with this plastic? Plastic is everywhere, not in the ocean, not only in the terrestrial part, or we can say in food also, fishes also, everywhere. So there are different types of the plastic. We can see polyethylene tetraphthalate, PET, HDPE, that is high density, low density polythene, polypropylene, polystyrene, and this contain different harmful compounds also, which can cause the dangerous diseases like cancer also, and some they can block the system, human system also, if it went to the through the food, or nowadays there are reports that our tea bags also contains the millions of, where we used to have a green tea with the deep things, that also contains more than the millions of the microplastic in that. So this slides particularly represent the plastic are relatively slow to biodegrade, with the serious health impact if accumulated in the plastic within the organism. So here you can see the small particles of the plastic eaten by the shrimps, and these shrimps will be eaten by the small fishes, and small fishes by the big fishes. How the plastic magnify in the small organism to the big organism like fishes, and that will be eaten by the human being. So this will be the biggest public health risk if we will not address this issue on time, because ocean is full of, now it's a soup of plastic. It contains millions of particles. Within a ml also will get the microplastic. So accordingly, what I personally did, in a couple of months, we have catch one fish. I was there for the marine environmental assessment study. So we have collected water sediment sample for more than 50 parameter. And in night, I just put the fisherman net in that. So we got morning a very big fish. That's a croaker fish, and I asked the local fisherman there, is this fish is locally available here, or generally you have seen of? So that fisherman says that we haven't seen this fish from last 3 decades, 30 years. So I got surprised, because this is a very precious fish, and what is in my hand is the gas bladder, we can say the gas slag, which is per sack price is more than the 10 lakhs in Vietnam. They are using this sack for the as a suture thread. So what I did, I have dissected all these fish, because they saw it's a rare fish coming in the Amba estuary near to the Dharamtar Port Alibag, and I have dissected in different part, intestine, muscles, and this one. So we have seen different types of microplastic inside that fish body, and this is which is characterized by the FTIR, Fourier transfer infrared, and the Raman spectrophotometry, that comes from that plastic having the different types like LDP, HDP, and PET, and the PS, polystyrene also. So it shows that whatever the fish we are eating in our plate is not safe for us actually. So further studies is ongoing, like whether their intestine also contains some toxic microbes or not. We will be doing the metagenomic study of that. But this initial results are really very shocking, and this is what happening in water, in marine water. This particularly slide represent the plastic hanging to the mangroves plant, and the plastic pollution interfering the mangroves ecosystem in the marine environment. With the continuous gathering of this plastic pollutant in the nature due to the inappropriate throwing of the plastic waste, undegraded accumulated plastic pollutant must interfere with the normal processes. It can change the biogeochemical processes and the natural processes which can hamper the biogeochemical process of particular ecosystem, because there are certain rivers which meets to the ocean, and the middle part where the tidal flux will be more, we called as a estuary, where the brackish water used to be there. If we throw, or we can say, the not only the plastic or the unregulated or partially treated sewage or the effluent, industrial effluent, so that will certainly create anoxic, like less oxygen or hypoxic conditions, which can kill the marine biota present in that particular marine ecosystem or the river. So what we have done, this is the part of diagnosis of the plastics and all. We came across two research paper which is very interesting. One of this research paper, the mechanism of biodegradation of polyethylene, was published in 1987, which says that if polythene is degraded, it will generate the carbon dioxide, which they have seen, and it can be biodegraded by the certain organisms, but it takes time. And the very recent one, which is published here, which is the left side of me in the slide, they have done the isotopic study and published this paper in January 2023, very recently, wherein they have studied 13 carbon labeled polyethylene to investigate whether the bacteria really consume the plastic and convert to the byproduct like carbon dioxide. So it proves that it can convert the polyethylene, it's a hydrocarbon, it will just degrade that and convert to the CO2. So we frame some objective. What next to that? Because this was the last report on the plastic mitigation by the Netherlands Institute of Oceanography. So here, what we have done, we have just fixed some objective to characterize, isolate the plastic degrading organism, which can be tested on the different plastics like high density polyethylene, low density polypropylene, and phthalate. And we have enumerated the carbon dioxide production by degrading the different plastic by the selected and the screened microorganism. And whatever the CO2 is generated by the degradation, it's a very slow process. We have monitored with the certain CO2 sensors, and then whatever the CO2 generated, we utilize that CO2 for the growth of microalgae. And after growth of that microalgae, whatever the biomass generated, that biomass we have used for the lipid extraction and transesterified to the biodiesel. So in this way, there will be the zero waste. Plastic is degraded. Whatever the byproduct generated in the form of carbon dioxide utilized by the microalgae, and then after that, the lipid is extracted to convert into the biodiesel. So on this, very recently, 6 months ago, we have filed one of the patent, a US patent, and the second paper is on underway. So what experimentally we have did here, we have collected submerged plastic in the beaches of Mandvi, Gulf of Kutch, Gujarat, and isolated this P1, P2, P3, P4 up to the 6 to 7 bacteria, and these bacteria are screened on the special agar. Hasnal bush agar, which is depleted with the carbon source, and only carbon source we have provided in the media was plastic powder, LDP powder, which is specially we have prepared up to the 500 micron size by the cryogenic grinder. And what we found, the only bacteria will grow there is plastic degrading, because there was no other source than the plastic as a carbon in the media. And after that, whatever the bacteria we got, we have characterized by the 16 as RNA gene amplification. We have sequenced, and then we have done the blast with the online databases of NCBI. We have characterized all the bacteria. And the most prominent bacteria we got is P2. Here we can see, because when CO2 dissolve in the water, it will form the bicarbonate. CO2 plus H2O, it will give HCO3 plus H plus. So this bicarbonate is more in the P2, 2.61 millimolar. And this is the modified MPN technique which we have developed in our lab, which shows this is a Hashnell broth, and inside there is a durian tube. Once the bacteria degrade the plastic, microplastic powder, it will generate CO2, and that CO2 gas will be trapped into the durian tube. And this is the principal component analysis graph, wherein we found the P12 bacteria. The code we have given different isolate, we have given different codes. The P12 bacteria having that capability to degrade the plastic within a 35 days. So next, this was our system we have just designed. It is a novel system so far. Nobody has tried this system in the world. So we thought to prepare this and see the, really is it working or not. This is a simple incubator. Inside there is a magnetic stirrer. On magnetic stirrer there is a Hopkin flask containing the Hashnell broth and the plastic powder, plus the very best efficient bacteria which can degrade, which we have screened earlier in the form of bicarbonate production. And this bacteria will degrade here plastic powder and will generate the CO2. And then this CO2 we are measuring with, this is the CO2 sensor we have. And we have periodically checked that how much CO2 in the ppm it generate, and that much CO2 we have given to the upper flask. It's we can see which having the light also, because the algae is a photosynthetic organism which require the water, sunlight, or we can say artificial LED light also we have used here, along with the CO2 as a major nutrient. In this way, the plastic is degrading, byproduct is also utilizing to form the algal biomass and convert into the different byproducts. So one of the byproduct is the biodiesel. Another byproduct also we have produced, that earlier said we have just filed a patent, and it is under the evaluation. So this graph represent like in 35 days how the growth of bacteria. Here we can see this is the special TVC graph, total viable count or total bacterial count. Here up to 21 days there is a continuous growth, but here the growth is more because the total viable count we have measured by the efflorescence with the fluorescent DAPI dye, which count the not only viable but the dead bacteria also, which binds to the nucleic acid of the bacteria. That's why up to 35 days the bacteria are more. And this is the total viable count. Here we can see from 1 to 28 days the bacterial count is more, and then it is deteriorating due to the nutrient depletion. So it's the only viable count, but it doesn't mean that that flask is not containing the dead bacteria. And this lower portion shows like optical density also increasing, just like total viable count of the bacteria up to the 30 days, and 35 days then it is declining. And this is the standardization in hours up to the 60 hours of that CO2 sensor. Then very first day or first hour the CO2 started what do you call, generating from the degraded product of plastic, and up to 60 days it went to the almost 5000 ppm. So here, very interestingly, we came to know that if we bypass the CO2, the degradation capability will be more. This is 2.3 gram in high density polyethylene, 2 in the low density polyethylene. And if we didn't bypass the CO2, so it may inhibit the growth of bacteria, because bacteria we use in the system is aerobics. And if CO2 we are not using for the algal growth, so that will give the feedback inhibition kind of, or we can say it will suppress the growth of bacteria. So this is a very important finding, and so far nobody has addressed this finding. So we are trying to communicate in a very reputed international journal. And this is the FTIRI, wherein the lower portion show the standard LDP, HDP, polypropylene, and PET graphs, and this is the treated one, wherein at certain groups we found there is a degradation, there is a gap, there is a change in the chromatogram of FTIR. And this is the scanning electron microscopic images. The down one is the standard plastic powders. It's LDP, HDP. We can see there is a very intact because the bacteria is not treated with. Where and in upper portion here we can see it is a treated when bacteria stick to the plastic, and it's there is a deformities in the plastic particle. That means it is utilizing the plastic as a carbon source and degrading the plastic. And this is the comparison of microalgal biomass with and without CO2. As mentioned earlier, that if we have bypassed the CO2, there will be the good degradation or a maximum degradation. If we don't bypass the CO2 from the flask, it will inhibit the growth of the bacteria. So likewise here, the algal biomass and the percentage of algal biomass is shown, and what is actually happening inside the cell also we have shown how the hydrocarbon plastic is degrading and generate the CO2 in a tricarboxylic acid cycle. So this also we have one of the slide I have not shown here as it is a IP issue, intellectual property, but I can mention here that we have done the whole genomics of this bacteria, wherein more than 31 genes we got, and we have represented on the circular map of this bacteria, and we have proposed the pathway of degradation of different plastic, because these genes means enzyme, because all gene product are enzyme. These are involved in that pathway which can degrade the plastic hydrocarbon to the CO2 and can be utilized for the algal biomass, thereby the biodiesel production. So here the algae we have used is a Chlorella vulgaris algae, which is easily can be grown and has the lipid content more than 34 to 35 percentage. So the down one is a Nile red staining. Nile red is a lipophilic dye. It is having the affinity to the lipid droplet inside the algal cells, and it will show the yellow to golden yellow fluorescence depend upon the intensity of lipid inside the cells. And then we have extracted the lipid and transesterified to the biodiesel. So conclusion, what we conclude on this, like the bacteria, the best bacteria we found from the Gulf of Kutch was the Pseudomonas mandosina. I have given the name Pseudomonas mandosina ABF with my initial 786, and we have already submitted to the NCBI, and we got the accession number, the all raw data, CROCA files, cake files, all functional and taxonomic annotation we have submitted internationally. We got the accession, and very soon we'll be submitting to the very reputed journal these findings, and the genes involved, the pathway involved will be proposed by India, and we got the opportunity to communicate that research. So this is a really, we are honored that we got certain result which having the capability to degrade the monster pollutant like plastic, and we can utilize the degraded product in the form of microalgal biomass to produce the biodiesel also. So the strategies to mitigate the plastic pollution, as you know, the reduce, reuse, recycle, renovate. This photo I have taken in one of my sampling near to the Alibag, wherein I saw this is a mangrove plant, and in high tide, lots of plastics, the shades used to come, clothes and all litters, and they wrap this mangrove. And sometimes the nematophore present in the biosphere, rhizosphere of this what do you call mangrove also. So that can kill the the mangrove, which is very essential for the marine environment to protect the coastal this thing from the tsunami and the coastal erosion of the beaches also. So I thought this is very interesting, that it is an eye opener that what human being is doing with the environment. It's really essential. Maybe if somebody from the university can take the cognizance the how important environmental science subject is, this for the and at the inception studies also they should involve this subject to the students at a higher level. Of course, every university UGC should make it mandatory that in India, every university coming under the UGC University Grant Commission should have the environment science department, which can really educate the people or disseminate the knowledge that what being a human being we are doing with the environment, to the animal, to the marine environment, or to the birds, avifauna and everything. This is really like a pathetic situation. The climate is changing, but people's mind is not changing towards the environment. The high time to change and to reciprocate. And definitely I would like to appreciate the Professor Praveen Meshramji, who is a very dynamic personality in Nagpur University, who has always taken the initiative, some new initiatives for the environment science subject. So basic this coming to this slide, the strategy to reduce, reuse, recycle, and innovate to promote the reduction of the plastic consumption through the awareness campaign and sustainable environment, encourage the reuse of the plastic product and packaging, the extent of their lifespan, emphasize the importance of proper waste management and recycling the strategies, highlight the advancement in plastic alternative. They need to find out some alternative for the plastic, because there is not a single day. Everybody, all professors listening to me must be knowing that our day is started with the plastic and ends with the plastic. Where we go, there will be certain atom in the form of plastic will be with us. हमारा मोबाइल क्यों ना हो या पेन क्यों ना हो या कुछ पॉलीथीन, जब सब्जी लेने जाते हैं तो वो भी, हम घर की थैली भी लेके हम मॉल में नहीं जा सकते, कि हमें पता है कि हम मॉल में जा रहे कुछ सब्जी या कुछ खरीदने के लिए, लेकिन हम अपनी बैग या थैली नहीं लेके जाते, क्योंकि हमें लगता है हम घूमने के खाली हाथ घूमे तो अच्छा है। लेकिन वो एक दो पॉलीथीन ही एनवायरनमेंट में बहुत बड़ा डिफरेंस क्रिएट कर सकती है। अगर ऐसी ही सोच यहाँ पे अभी 140 करोड़ जनता की होगी, तो आप सोच सकते हैं कि एनवायरनमेंट के साथ क्या होगा। तो ये बहुत ज़रूरी है कि हमें जो भी चेंज है वो खुद से ही स्टार्ट करना होगा। Think about the alternative. Here we can say to the no plastic straw, stop serving the drink with the plastic straw, no more single serving plastic, and refill reuse water bottles for the free, make single use items certified compostable, encourage biocharge extra for single use. So there are different strategies to avoid the single use plastic we have mentioned here. And as mentioned earlier, they are the five, right? Reduce, reuse, recycle, reuse, and repurpose. Repurpose, this meaning finding a new uses for the plastic items that cannot be recycled, such as using the plastic bottles or planters and the building material. So this can be the repurpose of the the generated plastic that we can use as a building material. Nowadays, people are using the plastic for making the road. They are mixing with the tar and making the road, so that it will be waterproof, will not form the the pit like now everywhere we can see in the monsoon season. And that is also done by the technology, also done by one of our system custom CSIR laboratory. So being a common layman, what we can contribute to the nature? We can always go for the beach cleaning campaigns. This is my campaign. I used to always, every Saturday used to have a very big team. You must be knowing the advocate Afroz Shah, who got the UN Champion Earth Award, first in India, later on our Prime Minister Narendra Modi who got that. So he's one of my friend who always accomplish these things, and try to make awareness about plastic and other pollutions in the beaches. And every Saturday he used to be there for the Warsaw beach cleanup. So this is one of the photograph of beach cleaning. There are different placards to spread awareness that how we can. Here this very old photograph of my, where I was just picking the trash of plastics and other thing. तो हमें हमारी तरफ से जितना भी हम कर सकते हैं एनवायरनमेंट के लिए, है हमारा दायित्व कि हम करें, और उसके संस्कार हम घर से ही शुरू करें। This is a very interesting slide I just put to show you. In 2020 or 19, I met to the Nobel laureate Sir Richard Robert, and we had one meeting wherein while having tea, he made one sentence that we need more science in politics and less politics in science. This statement fascinates me because everybody knows that how the politics sometimes interfere in the science also in the form of funding and the other things. Everybody knows that, but there should not be any interference of the political science in the science. Now you know that the politics is everywhere. For taking the darshana, we have to pay. We'll get the VIP darshan then in temple and big spiritual places also. But the science should be untouched with this kind of the thing. Then and then we can really innovate or renovate the novel thing which is required for the well-being of the mankind. And this is the way we can disseminate the whatever the knowledge we gained into the laboratory. And this particular slide of my very last year, I guess, the Doordarshan has invited me for the person to they made a program in the World Ocean Day. And these are some of our research published in the press and electronic media. That can be the media to disseminate the science generated in the laboratory should not be limited to the laboratory, but the common people also come to know that what kind of the research you are doing, and that research how far will be useful for the common man or can spread the awareness among them to keep the environment sustainable, which is the theme of our this refresher course. How we can sustain the environment, that's very important. And this is my team. I have 4 PhD students. They are working on the different aspect. The guy beside me is the Harshal Jadhav, who is the CSIR SRF. He's working on the plastic degradation. This is Mr. Avinash Aushank. He's working on the bioprospection of marine fungi. He's creating drug from the marine fungi associated with the mangrove nematophore. And this girl is Ashna Moga. She's working on the bioremediation of heavy metals, especially chromium, by the marine microorganism like Bacillus ankylosis. This bacteria she found it out, and recently she is going to submit. One of the students submitted and awarded the PhD, and 2 more this year going to submit. And these are the different birds you can see. This is captured by all photos by me. I have especially mentioned there to see how beautiful is the nature, especially the marine nature, because I am concerned with the marine. But the environment is only one. Ocean is a part of the terrestrial and the other environment. These three birds are the ibis. This is the black-headed ibis. This is the red-napped ibis. This is the glossy ibis. Because we used to do the avifauna study also. Sometimes it's really important for us to document the kind of birds present in that ecosystem. And this is the swamp hen. So many birds are coming from the, they are migratory. They are coming from all the way from the Europe and here, and the flamingo sanctuary is also here. So we need to take care of this. There was one experience. One day I went for we have the Lokhandwala Lake backside to our institution. I went to just capture some photo in the morning. I saw one of the bird was struggling whole plastic. That bird name was cormorant. Jala आपण पानकोंबडा म्हणतो. It just made a dive into the lake, and it came up with plastic which is wrapped towards his beak. He was struggling to get out of that. 1 hour, 2 hour, I was helpless. I was because he was the bird was in the lake. तो he was struggling to वो कि कैसे उसको निकाले, वो struggle कर रहा था. और मैं भी 1-2 घंटे वहां रुका कि मैं तो कुछ मदद नहीं कर सकता था. लेकिन दूसरे दिन जब मैं मॉर्निंग को आया, so the picture I saw was very shocking. I saw the dead body of that bird, and the plastic was wrapped towards his beak. So this is really, really pathetic. We need to wake up while throwing the trash on road, beside our home, or everywhere. The trash should go into the dustbin, and we have to make sure that dustbin is also going to the municipal corporation vehicle at the proper destiny or not. Then and then, as we can say the good citizen of India, we are contributing toward the sustainable environment. So this is from my side. If anybody having any question, they are welcome to ask me. Thank you so much.
Now I would like to request participant, if you are having any query, please interact with our expert.
Yes, sir. Dr. Abhay, sir.
Yes.
Good afternoon, sir. I was your student, sir.
May I know your name,
yeah.
please? May I know your name, please?
Dr. Amit Kalwande.
Oh, I mean, yeah. Tell me.
Sir, actually, I want to ask one question.
Yeah, go
Sir,
ahead.
actually, you just recently said that plastic roads, means that plastic road, ये solution हो सकता है. But I
Yes.
think, sir, that may leads to the microplastics because plastic usually means that the duration of plastic is from 20 to 100 years. But most of our roads actually we have the duration of that road. We know that actually it may be up to 10 years, and most of the flood and that other problems that will carry that microplastics to environment only. So what do you think that is this that means permanent solution or not, sir? That plastic roads are good enough to have that particular sustainable development के लिए?
Yeah, it's a very wonderful question. What I came to know, I have little information about this research. One of the CSR laboratory, Central Research Road Research Institute, they have done some study. They have blended the plastic pieces with the tar, and they have
Yes,
made sure that it will not, resuspend to the water or any solution once it is
yeah,
implemented on the road. So this, then and then, because we used to see ethically, like it should not be a problem creator in future. And as you said, that it is not a permanent. Nothing is permanent in the world. But
yes, sir.
how much the lifespan we have, so we should take a precaution to make a sustainable technology which will not create a problem for the future generation. This is what we can do the best.
Yes, sir.
Now we can see in the name of development, lots of trees and everything is cutting, but nobody is doing the audit of that whether they really on behalf of that other they are planting the plant or not.
Yes,
आपल्याला रोपण करता पण आलं पाहिजे आणि रोपण करता करता संगोपन पण करणं खूप आवश्यक आहे. आपण रोपण हरवर्षी एकाच जागेवर पण मी वृक्षारोपण करणारे लोक बघितले जे फोटो काढण्यापुरतं तिथे झाड लावून देतात. मग त्या झाडाचं काय होते कोणाला माहिती देवच जाणे. आणि दुसऱ्या वर्षी सुद्धा तिथेच एक झाड लावलं जाते प्रमुख अतिथीच्या हाताने आणि एक फोटो खिचला जातो. तर त्यामुळे रोपण सोबत संगोपन पण खूप आवश्यक आहे. ज्या प्रकारे आपण जिस प्रकार से हम अपने बच्चों का पालन पोषण करते हैं, उन्हें सर्दी खांसी भी हो जाए तो तुरंत डॉक्टर के पास लेके जाते हैं, उस प्रकार की भावना जब तक हमारे मन में वृक्षों के प्रति, ट्रीज के प्रति नहीं आती, तब तक मैं नहीं समझता कि जो भी हम मैनेजमेंट प्लान, एनवायरमेंट सस्टेनेबिलिटी की बात कर रही है वो सही मायने में महत्वपूर्ण होगी. पेपर पे
yes, sir.
लिखा कुछ अलग होता है और वास्तविकता अलग होती है. तो हमें हमें अभी उस पे ही निर्भर ना रहते हुए खुद को इन्वॉल्व करना पड़ेगा इस चीजों में. देन इन दे सही मायने में हम पर्यावरण की रक्षा कर रहे हैं ऐसे मुझे लगता है.
Yes, sir. Sir, रिसेंटली मैंने वही एक्चुअली आर्टिकल पढ़ा था कि जो प्लास्टिक रोड है दैट मे लीड्स टू द माइक्रो प्लास्टिक बिकॉज जो फ्लड वाटर है या फिर रनिंग वाटर है उसके जो डिग्रेडेशन होगा प्लास्टिक का दैट लीड्स टू
I'm
द
agree
माइक्रो
with you.
प्लास्टिक.
Who invented this polymer, so-called plastic? Also,
यस,
that time I have not thought that this will be the problem in future. Huge
या,
problem, right? तो हर चीज की
यस,
एक सीमा होती है और एक लिमिट होती है, एक मर्यादा होती है. और जहाँ पे मर्यादा का उल्लंघन होता है तो वो वैसे ही इसका सब लोग फिर सर्वनाश की ही दिशा देखते हैं.
यस. थैंक यू सर.
You are welcome.
थैंक यू, थैंक यू.
Next participant, please.
Good afternoon, sir.
Very good afternoon.
Sir, my name is Dr. Vijan Kumar Shil. सर
Yeah.
मेरा कोई सवाल तो नहीं है बट आई कैंट रेजिस्ट माई सेल्फ टू से दैट दिस इज वन ऑफ द बेस्ट सेशन वी हैव इन आवर रिफ्रेशर कोर्स. ऑलमोस्ट ऑल द पार्टिसिपेंट्स विल बी एग्री विद मी. थैंक यू सर, काफ़ी कुछ सीखने को मिला सर. वेरी नाइस. थैंक यू
Thank you.
सर.
Thank you for your appreciation. Though what I want, like, I don't want a clapping or standing ovation or anything, but if you can plant one tree and take care of that, once it is grown, no, it will give the, the useful fruit to everyone. This will be the biggest appreciation for me.
Yes, sir. Of course.
I would like to say one thing, sir.
Dr. Rajana, please proceed.
Sir, can we do plastic biodegradation on large scale?
Yeah, we are trying to make a prototype lab scale And this can be done. As I told you, the bacteria I have presented here is Pseudomonas mandocina can degrade the plastic in 35 days, but certain quantity in lab controlled condition. So we need to check it whether it is really happening outside. Lab to land of transfer of technology, we have to do it then later. So we are planning for the small pilot scale plant where we'll be throwing this bacteria bucket full of bacterial broth on the plastic, and we'll be watching what will happen to that plastic. So this can be done, but on R&D basis. Until it is not done, we cannot means recommend this bacteria in the environment.
Okay, great, sir. Thank you, sir.
Thank you.
I'm having two questions, sir.
Yeah, very good
Question,
afternoon.
hello. Sir, this microplastic will cause a change in the DNA of the microorganism, sir. So that is the DNA of the marine organism, sir. This
Yes,
microplastic.
yes. As I showing that fish having the all parts, in the
Yes, sir.
intestine, in the muscle, everywhere the microplastic is there. So it can have, damage the internal organs, even human being organs also. And it contains like polyethylene tetraphthalate and polystyrene. Some, it can be a carcinogenic also. It can cause the cancer also sometimes.
So it is better to avoid fishes and also for consuming purpose.
I didn't get you. Can you just speak loud?
No, sir. If we are a non-vegetarian, it is better to avoid fishes. I'm asking
Yes, yes, that can
for
be,
consumption.
yes.
Next participant. Again, anybody would like to ask any question?
Please feel free to discuss. Do not worry about, because all our professors and the doctors, we had to collectively discuss for the sustainable environment. Like, what I can do, I am always there for any collaboration or anything. Dr. Professor Praveen Meshram sir knows me very well, and he has supported me when I was the student. So I'm really thankful to him, like, that he still stick to that things, and like serving the nature and the environment.
So you were mentioning that everyone should take care of our ecosystem, that is our nature, no, sir?
I'm
Hello?
unable to hear your voice. Can you just?
Sir, is it okay now?
Yeah, tell me.
Sir, you were mentioning that we have to be responsible for the environment. We have to take care of it in all the possible means, no, sir? But this
Yes,
itself is creating a great disturbance for us, sir, mentally. Because if we are responsible, the persons outside us are not at all responsible. So it is creating a lot of stress inside us.
lots of.
No, sir. Everyone is not at all thinking about the environment. Just we will be only thinking means we are, if we plant even a single tree also, they are cutting it or they are using some sort of disturbance to the plants. So it is totally disturbing us.
Yes. No, what I said, madam, means what is my, it's not only the plant that we can grow and save the environment. Environment itself is a very strong to rejuvenate, so we don't need a human intervention in the environment. That is enough for the environment. And the second thing, the seed of thought should be buried or sowed into the different minds. Now you people are teaching to the many students. Okay, so there also there is a platform to give the discoursement on the environment. I remember when 2019, 1916, Environment and Science Technology Minister Dr. Harshvardhan visited to my lab. He spent whole day with me here and discuss about our research. So I requested him to pass one of the message to our president, who is the Prime Minister of India, Dr. Mr. Narendra Modi, that being a religious country, we have different platforms here. Hindu, Muslim, Sikh, Isai, and all Dharma Guru used to give their own pravachana, which is followed by blankly by the people who are the believer in the particular religion. So I requested why not use that platform for discoursement on the environment. उस वैसे धार्मिक प्लेटफॉर्म का अगर हम पर्यावरण पे प्रवचन के लिए उपयोग करते हैं और वो अगर उनके उनके धर्मगुरु वो प्रवचन देते हैं तो लोगों पे इसका बहुत अच्छा प्रभाव पड़ेगा और वो लोग बिल्कुल यानी विश्वास करते हैं और वैसे करेंगे. उनको बोला जाता है कि 10 बार पूजा करो, ये करो, तो वो करो, या तो 2 पेड़ लगाओ बोलेंगे तो वो लगाएंगे, बिल्कुल लगाएंगे, जिसके कारण सृष्टि का संरक्षण हो सकता है. तो उन्होंने वो बहुत ही अच्छा लगा उनको, टर्मिनोलॉजी अच्छी लगी, पर्यावरण पे प्रवचन, लेकिन उसके बाद फिर उस प्रकार से उस पे इम्प्लीमेंटेशन हुआ या नहीं है उसकी इंफॉर्मेशन मुझे नहीं मिली. लेकिन अपने हाथ में इतना है कि आपको रुकना नहीं है. आपके जो विचारों के बीज है, जो थॉट्स, जो सीड ऑफ थॉट्स है, वो आपको हमेशा सो करने हैं. उसके क्या बोलते हैं, उसको बोना ही है आपको यंग माइंड्स में. जो आपके स्टूडेंट्स है, जो भी है, जो भी प्लेटफॉर्म मिलता है, शायद 10 में से 2 लोग पेड़ लगाएगा और 2 पेड़ में से 1 पेड़ भी बच गया है तो आपका मिशन सक्सेसफुल हो गया है. मुझे ऐसा लगता है जो एनवायरमेंट सस्टेनेबिलिटी का है. दिस इज व्हाट आई कैन से.
Thank you, sir.
Dr. Abhay, I would like to request you to share your that project regarding the lab to land.
Okay, sir.
I would like to request you to highlight that theme of your project which you have handled, where you have worked as a scientist under Dr. Sudhir Meshram sir.
Sudhir Meshram sir.
Regarding lab to land.
That is the very old one, sir. Actually, I have done the 2 PhDs. You know, one PhD was with Dr. Sudhir Meshram, and then in Neri, Dr. Tapan Chakraborty, sir. So there, I worked on the climate change actually.
Okay.
So that was a CO2 sequestration project. But right now, I do not have that one to just show that slide here. But it's a long back. I worked on that, but I have everything. Like means maybe we can discuss that thing also.
I like your thought regarding any science subject or any institution or any university should be away from the political interferences.
Yes, sir. That's what I told. Like this particular slide represent the statement for the same. We need more science in politics and less politics in science. Sir, this Sir Richard Robert is a postdoctoral student of James Watson and Crick, who got a Nobel Prize for the double DNA helical structure in 1963. And he also got the Nobel Prize in 1993. Robert Richard. He is a postdoctoral student of James and Watson, who represented world's first DNA structure.
Very nice. Even I'm very much thankful for your for giving the suggestion in order to start the PG department in every university, which is the need of.
That is very essential. Very soon, I am going to meet a UGC chairman also. Maybe I can put this thought in front of him, and how important is this? Like
when in our Nagpur University, we are lagging behind. On the other hand, small cities just like Nanded, Aurangabad, Jalgaon, Kolhapur, Solapur, they have started their own department of environmental science.
Yes, this is really surprising, sir. Asia's biggest national environmental engineering research institute is there, NERI, in Nagpur, and university don't have environmental department.
Yeah.
This is really, but you have established in Sevadal College, so that is really appreciating. Like you are doing your part very enthusiastically and very dynamically. So that's what this vision is very essentially at university level also.
Our institution is the first private institution where we are running UG department, PG department, we are running research activities. And this is totally a unique case in the history of the Nagpur University.
Right. Sir, along with this talk, I would like to invite each and everyone present in this talk to my laboratory to personally see whenever they are in Mumbai, or if they want to avail some facility. So we have everything. We have microbiology, ecobiology lab, genetic, everything. if they want to avail that facility for their students, just let me know. I'll be in touch with them. So we can do some collaborative research also. We can publish papers also. So everybody is most welcome. The only thing what I expect from everyone that we have to, it's a high time we have to serve the nature, we have to serve the environment for the sustainability.
Yes, yes, very nice. I'll request all the participants, please take the advantage of this opportunity which Dr. Abhay is providing to you.
My details are there in the slide. You can see with the mobile number, email, everything.
Yeah, yeah. So now I would like to request Mr. Kereketa, please propose the vote of thanks for this
Okay.
beautiful presentation
Yeah.
made by Dr. Abhay.
Yes, sir. Yes, sir. Okay, sir. Okay. At very first, I would like to thanks Dr. Mesram to call just like Dr. Phulke, sir, and he arranged like this session. And what thanks we render for Dr. Phulke for his amazing and challenging task. We I don't have trying to find such type of give him thanks. He enlightened us about the high and low degradation, and he is special focused about the reuse, reduce, recycling, and the innovation, etc. एंड आई थिंक अब से हम कैरी बैग लेकर बाजार जाएंगे ऐसा मैं विचार कर रहा हूँ. And once again, on behalf of UGC, HRDC Nagpur, and all from all participants, and we are very thankful for this very, very informative and very interesting session. Thanks a lot, sir. Thank you, sir.
Thank you, sir.
Over
Thank
to,
you so much.
yeah, over to Ms. Ram, sir.
Okay, Abhay. I'll
Yes,
keep in touch with you, huh?
yes, sir. Thank you. Thank you for inviting me, sir.
Yeah.
Thank you.
Okay.
Thank you, sir.
Shall we take a leave then?
Yes, sir. Thank you.
Bye.
Bye.
So shall I take your attendance first? Then I'll give you the next day instructions.
Yes, sir.
Roll number 1, Dr. Dipti Malviya.
Yes, sir. Present.
Dr. Rushali Parkhi.
Present, sir.
Miss Neetu Amlani.
Present, sir.
Neerav Pandya.
Present, sir. Present, sir.
Mrs. Minal Rajdev.
Present, sir.
Then Dr. Suparna Murkute.
Present, sir.
Dr. Amit Kabande.
Present, sir.
Dr. Archana Mahakalkar.
Present, sir.
Dr. Vinod Dongarwar. Not there, no? Dr. Arvind Somnathe. Dr. Shruti Gharde. Dr. Shruti Gharde. Any problem in connectivity? Dr. Meenakshi Kamble.
Sir, Pallavi Deshmukh.
हाँ
Present, sir.
हाँ ठीक है ओके. डॉक्टर मीनाक्षी कांबळे.
Present, sir.
डॉक्टर शील.
Present, sir.
हाँ विवेक माणिक.
Present, sir.
डॉक्टर राजेश सोनकुसरे.
Present, sir.
डॉक्टर विशाल दौलतकर. डॉक्टर स्वाति यादव.
Present, sir. Vishal Developer.
हाँ डॉक्टर स्वाति यादव. मिस्टर Kerketta, you were there.
Yes, sir. Yes, sir. Present, sir.
Yeah. You're from Raigad.
Yes, sir.
Miss Kani Mozi, always present.
Present, sir.
Yeah. Dr. Shikha Avasti. Shikha Avasti. I think she has left. Or she is also facing problem in connectivity. So now we'll be meeting on Monday at 10:50 and we'll be having the first session of Dr. Shalini Dani. Again, she's one of the scientists from NERI and she will be delivering her presentation on the degraded lands of all over the world. Then we'll be having second session of Dr. Mahendra Patil. Again, he will be heading the Department of Solid and Hazardous Waste Management and he is going to deliver his presentation on the recent issues and the challenges in solid and hazardous waste management. Then after lunch, we'll be having third session of Dr. S.R. Verma. She's working as a associate professor, Department of Zoology at Anand Niketan College, Varora. Recently, she had joined to Varora. Earlier, she was also in NERI and she is going to deliver her presentation on environmental impact assessment with special reference to her in context with our country, India. So this will be the timetable of Monday. Now, regarding the brief introduction and the vote of thanks of Dr. Shalini Dani, who will take the initiative? Dr. Dawlatkar पण तर झालाय ना. Dr. Swati Yadav, roll number 19. Swati Yadav. Otherwise, shall I request Miss Kanimozi?
Yes, sir.
You will have to take care of the Shalini Dani.
Yes, sir.
Dr. Shalini Dani. She is a principal scientist of NERI Nagpur. And her topic and her regarding her brief biodata, I'll be forwarding to your group.
Yes, sir. Okay, sir.
Then regarding Mahendra Patil also, you can conduct his proceeding.
Okay, sir.
Dr. Kanimozi. Dr. Mahendra Patil also, I'll be submitting his biodata in our group. The topic
Okay,
of
sir.
his presentation is the recent challenges in solid and hazardous waste management.
Okay, sir.
This is how you will have to take care of both the scientists. Introduction as well as you will have to propose the vote of thanks. Then I'll be shouldering the responsibility of the third session to the Dr. Shikha Avasti.
Yes, sir. Sure, sir.
Okay. So shall we take a leave? You would like to ask any question, any queries? Have you submitted all your documentation regarding your relevance certificate and all?
Yes, sir.
Anybody is having any problem?
Sir, from Monday we would be having our seminars, na?
Yeah.
Sir, from what time? So, are we going to have the third session also?
Just a minute. Let me get you the time paper, I think. Yeah. Oh, yeah. Just a minute. First session will start at 11 o'clock.
Okay.
Then it will end to 12:30. Then immediately, without taking any break, again we'll be joining to 12:30 and we'll try our level best to end up at 1:45.
Okay, sir.
And then you take 1:45 to 2:15 your lunch break.
Okay.
And then then I have already requested Dr. Verma madam. She is going to start at 2:30 only. And 2:30 to 3:30. And then from 3:30 onwards, we'll be having the seminar.
Okay, sir.
3:30 to 4:30. And regarding seminar, first 4 roll numbers.
Sir, madam had forwarded first five roll numbers.
Okay, first 5. Means roll number 1, Dr. Dipti Malviya. You will have to get ready with your seminar. Then Dr. Rushali Parkhi. then Neetu Amlani, then Dr. Nirav Pandya, and Dr. Mrs. Minal Rajdev. त्यांनी 5 पाठवल्या म्हणतात. So we'll be starting your seminar at around 4 o'clock. And then 4 to 5, we'll have to finish out the seminar. And then next day, or Tuesday, again we'll be starting our seminar at 2:30. Now, is it okay?
Hello, sir. Is there any last date of submission of PPT? Uploading the PPT.
Tomorrow, na? Tomorrow up to 6 o'clock.
Sir, what's beyond getting you?
You can submit your PPT up to tomorrow in evening hours. Up
Okay,
to 6
sir.
p.m.
Okay, sir.
Yeah.
Sir, we had no seminar on Tuesday. The schedule which Preeti Dharmik madam had given us, na, there we didn't have any seminar on Tuesday.
Why? Tuesday we have kept seminar.
I don't know,
सर
sir.
24 ला सेमिनार आहे, त्यानंतर 26 ला आहे सर. मग
हाँ, ऐसा बोला था. 24 के बाद सीधा 26 को. There is no seminar on 25th.
Listen, yes, seminar will be on Monday.
Yes.
Sir, there must be some confusion. I'll talk to her and I'll communicate to you immediately.
Okay, sir. Thank you, sir.
Otherwise, I'll put a message to in our group.
Okay, sir. No issue. Thank you, sir.
Whatever the timetable which I have prepared and I have forwarded, under that what we have prepared, will be having seminar on Monday of 5 participants, 1 to 5.
Directly on Wednesday.
Monday on, that is on 24th July.
Yes, sir.
At 3 o'clock.
Yes,
Then
sir.
again we'll be having seminars from 2:30 onwards. 2:30 to 5 o'clock. Again, this roll number form from 6 to 12. The next day on Wednesday, again there will be a seminar from 12:30 onwards only. Then on Thursday, there will be a one separate webinar. This webinar they are going to arrange by Staff Academy College. That is in between 4 to 5 p.m. Before that, there will be a presentation from Gondwana University. Dr. Mahendra Thakre, he will be delivering his speech, his presentation. On Wednesday, there will be a presentation of Dr. Sanjay Thorat, who is heading the Department of School of Environmental Science at North Maharashtra University, Jalgaon. Then on Tuesday, again, there will be a presentation of Dr. Mr. Kaushik Bhattacharya. He is going to give a presentation on role of media in environmental education. Again, I'll keeping all regarding your seminar. I think there are some overlappings is there. So I'll talk to the Preeti madam and corrected timetable I'll put in your group. So shall we leave today's meeting? Yeah.
Sir, excuse me, sir. Sir, I didn't submit my abstract on 18th, sir. The link was not there on 19th, sir. 18th I didn't submit, so I thought of submitting it on 19th. But 19th, the link was removed from the website, sir.
Okay, I'll discuss all these issues with the concerning authority of the Staff Academy College.
Okay, sir. Okay, sir. Thank you, sir.
I'll talk to the Dr. Dharmik madam, Preeti Dharmik madam.
Okay, sir.
Regarding your submission. So we'll be meeting on Monday.
Okay.
At 10:45. So,
Yes,
shall we leave the today's meeting?
Okay, sir. Thank you, sir.
Okay.
Sir, we can have one discussion with you, sir. Sir, excuse me, sir.
Kanimoji, come on.
Sir, kindly allot some time for us, sir. I would like to have some discussion with you, sir. Regarding general topics, sir. Regarding EVS subject, sir.
Yeah. Come on. Do you have any suggestion?
Sir, I will prepare one list of questions, sir, which I need clarification from you, sir. General things regarding EVS, sir.
Okay. So you will be forwarding on my email ID or what?
What, sir?
Where you are putting all these questions.
Sir, allot one time, sir, so we can discuss in group itself, sir. No issues. General things regarding EVS, sir. We can ask others also if they wanted to discuss something with you, sir.
On which topic?
Sir, regarding general topic which is relevant to EVS only, sir.
Yeah, please. Ms. Kanimoji, do you want to ask regarding seminar? Do you want to ask anything regarding seminar?
Sir, just one thing, sir. Sir, whether the microplastics are present in the air itself, sir, it is causing air pollution too. Whether my voice is audible?
No, no.
Is it okay now?
Yeah, please.